So how did you get to that other?
I went to camera controls.
Okay.
Which is
Streaming controls. Show camera controls. Oh.
hi. So we're trying to stream.
Oh, it's working. We came up.
Did it? Yeah, sometimes there's a little bit of a delay when you
the
start.
audience.
It's streaming
The audience.
Oh yeah, let me switch the cameras to you.
Huh.
Yeah, sometimes there's a little bit of a delay. I don't hear myself talking. Be all set.
Okay, cool.
Okay, cool. Yep. Let me know if you guys have any more problems.
Thanks. Yeah, thanks. I
Thank
appreciate
you.
it.
Okay, thanks everybody for joining us for the postdoc talks. We're going to have four. Two are going to be pre-recorded and two are going to be live. And if you're watching on YouTube, you can just type questions in the chat and I'll read those out at the end of the presentations. So let's get started with Emily's talk.
Hey everyone, so I am Emily Reed. This is probably a lot of your first time seeing my face, and vice versa. I'm a postdoc here with the Global Change Center in the Invasive Species Working Group, and I started last week. So I'm really excited to be sharing a little bit about my dissertation research from NC State today, and hopefully connecting it a bit with my position here at Virginia Tech. So, I'm going to be telling you about the landscape genetics of an anthropophilic invasive mosquito, Aedes albopictus, or the tiger mosquito. But first, a little context. So we know that urban areas have grown immensely in the past, and it turns out that they're not projected to stop growing. So we're seeing unprecedented amounts of land conversion from vegetated, natural, or barren areas to impervious surfaces. And that's kind of demonstrated here from a paper from PLOS One, where we see the Southeast United States, and these researchers projected that urban land cover would increase by anywhere from 100 to 192% by 2060. And this presents, from a research perspective, a kind of unique opportunity, because urban areas are kind of cool from an ecological and evolutionary perspective. They're microcosms for observing species responses to disturbance in situ, seeing how they evolved and how they adapt. But from an applied perspective, it's also important to research ecology and evolution of species in urban areas for management. So mitigating the negative impacts of global land use change and conserving species of interest, and maybe controlling invasive species. And as I found out during my research, urban areas are really kind of the center for a lot of invasions and are dominated by invasive species. So I wanted to look at some of these patterns and processes for my mosquito using a population genomics approach. And population genomics is a really fun, new, kind of powerful tool to observe species changes at fine spatial scales. So looking at really small differences, genomic differences, to understand genetic connectivity, gene flow, and evolution. So again, my focal species was Aedes albopictus, the tiger mosquito. This is an important species, not only because it's invasive, it's a vector for disease, such as dengue fever. It's also a huge nuisance, that might be primarily what we think of when we think of mosquitoes. And they're really omnipresent across the landscape. And so today I'm going to tell you a little bit about my research, specifically using a landscape genetics approach to look at how urbanization affected fine-scale patterns of genetic connectivity and gene flow in a population of Aedes albopictus. And I conducted this at a pretty fine spatial scale, at the county scale. So Aedes albopictus is native to Eastern Asia. It really started expanding in the 1980s with the increase in international shipping trade. And now they're pretty much well distributed across the eastern and central United States. They're invading and expanding in Europe. And the reason why they're so good at invading has to do with their natural history, specifically their oviposition behavior. So these mosquitoes lay their eggs in small, ephemeral, still pools of water, and the eggs actually require a drying cycle before they hatch. So in natural environments, this would be things like tree holes, which I'm looking into here. But you can see how this behavior translates really well into human-dominated environments, because we have lots of artificial containers, things like bird baths, but also trash, refuse. I've been moving, so I had this big tarp to cover my stuff in the truck, and it rained a lot, and I didn't do anything about it, and I just created a little biota of lots of mosquitoes. I found like 3 different species, which was kind of cool, but I also have a lot of bites. So with this information, I predicted that urban and suburban areas with high densities of artificial containers would facilitate gene flow in Aedes albopictus at fine spatial scales. So we know this is important for international movement, but what about locally? And so to answer this question, I'm going to briefly tell you about my genomic approach, which is double digest restriction enzyme associated DNA sequencing. It's a form of reduced representation sequencing, so I'm kind of randomly pulling bits and pieces out of the genome to sequence and compare between individuals. I just looked at neutral markers, so we're looking at kind of the standing genetic variation due to drift and gene flow, not necessarily adaptive. And all things told, I sampled 42 sites, sequenced 276 mosquitoes, and from about 3 million genetic variants or SNPs, filtered them down to about 1,300 that I used in my analysis. My study area was Wake County, North Carolina. You can't really see it here. I tried to do like hokey colors for Blacksburg, that's the dot in Virginia, and NC State colors for Raleigh, which is the center of Wake County. And Wake County is actually a pretty interesting place to study this. It's right in the middle of Aedes albopictus's range here in the United States. And it also has a very interesting spatial pattern of urban and rural areas. So Wake County is rapidly growing. Raleigh is kind of there in the center. The population has doubled since 1990. And there's this interesting patchy distribution. So that one black area in the west is Umstead Park. So that's a preserved area of forest. But otherwise, we're seeing rapid expansion. And I chose to use development as a proxy for urbanization. So I don't know if you all are familiar with the USGS's National Land Cover Database, but they have 4 categories for development, which are going to be your urban and suburban areas. And these cover about 38% of the area of Wake County. I'm going to tell you a little bit more about these classifications because they will be important. 21 here is going to be your open space development. Those are mostly green spaces with some infrastructure, some buildings. It's going to be like you see here, like your fields, baseball fields, parks, golf courses. Then we have low intensity development, which are going to be mostly residential areas, single family households that are fairly spread apart from one another. So big backyards, trees, kind of like your older neighborhoods. Medium intensity development, which is 23 here, are going to also be single family households, but they're much more densely packed together. So think about, kind of the classic suburb. And then finally, we have high intensity development, just kind of everything else, your commercial land, your apartments, industry, business, etc. And so to look at how these specific features impact gene flow, I used this really cool tool to optimize resistance costs based on my observed genetic distances between the 42 different sites that I sampled. And so it's this iterative optimization algorithm that fits the cost distances against the observed genetic distances to find like the best ratios of costs. So again, these are all relative costs. And what is the kind of baseline is anything that's not developed. And this is what I found. So to distill it down, the lowest cost form of development was low intensity development. So spread apart houses. The highest cost was medium intensity development. So packed together suburbs. And this tells us, one, that type of development is going to impact patterns of gene flow in Aedes albopictus as we see development. It also tells us that as all of our undeveloped lands become developed, that low density, diffuse household is going to become the de facto baseline and start facilitating gene flow. So, once there's no more forests left, then those areas are really going to see a lot of movement and potentially, spread of adaptive genes between mosquito populations. And so I wanted to apply this. So I'm actually working with the different towns and municipalities of Wake County to look at their development plans in the near future to identify areas of likely growth. And what I found is that 30% of the undeveloped land in Wake County is planned to be developed by 2050, so a good portion. So I'm hoping that my work can help inform and be used to design policy and make planning decisions to prevent, expansion and growth of mosquito populations. And this really relates to my work here at Virginia Tech with the Invasive Species Working Group, because I'll be conducting research kind of at the interface of invasion biology and invasion policy across taxonomic groups and also across spatial scales. And if you have any questions, I'd love to talk to you all more, either about this work or what I'll be doing here. Thank you. Are we doing
No
questions?
questions online yet? Just go ahead and call on people.
Oh. Yeah.
Thanks, it's really interesting. I guess when you say policy, I'm curious, do you see most of the policy implication at the household level? In other words, whether through carrots or sticks motivating different household behaviors where we might see pooling, or is this more at the urban form scale, or what scale do you see that happening at?
So in Wake County specifically, mosquito control is done by private businesses. And so it's up to the individual household whether or not they want to use a spray or something. However, because Wake County is the home to the capital, right, Raleigh, we do have a lot of urban entomologists and policymakers kind of concentrated here, and they can influence, especially within the county governance, what those companies can and cannot do. So one thing that I haven't been directly involved with, but the lab that does a lot of the mosquito research has been involved with, is working with both those private companies and some of the planning groups in Wake County to think about how we can use control methods to protect whole neighborhoods with the minimum amount of spraying. So it's pretty cool, kind of the neighborhood effect of if you spray one house, does it help the other neighborhoods the other houses? And so, I don't think that there's going to be actual policy in Wake County for mosquito control anytime soon, but I think that there is enough kind of conversation being facilitated by our institution between these groups that they could work together to be a little bit smarter in their control. Yeah.
Do you think, you may not have an answer, but in the cost correlations with the says colonized and invaded, that they have different climate and environmental
Yeah.
areas, like more arid areas?
Right. Yeah, that's a really great question, and one that I think, struggled with as a PhD student. And I think that yes, the most important factor in determining these resistance costs, from what I can tell, is going to be the configuration of those different urban categories in a landscape. And anywhere where you have kind of like that centrally located urban area that's experiencing sprawl, I think that you're probably going to see, if not similar ratios, at least similar orders. So climate could be important, especially if we think about places where urban areas actually protect mosquitoes from cold climates. So it's like the urban heat island effect, and mosquitoes can kind of be sheltered there, like up in New York. That'll change things. But I think, across the East Coast, and I think a little bit further west, we'd probably see similar patterns. And this also sets up a good way for other people to do this research, because as genomic methods become less and less expensive, this is a kind of analysis that a lot of different towns could do. All right, thank you all so much.
Thank you.
Should we just close out of this?
Okay. I have a request to dim the lights so that people could see the screen better from YouTube. Not sure. What's that?
Can they hear?
I think so. They were complaining about the lights, not the audio.
Jeremy, you said Tadhg next, right?
Yeah, let's put him next. I'll try to find light.
Hello everyone, and welcome. My name is Tadhg. I am a postdoc in the Kerry lab and the Thomas lab, and today I'm going to talk to you
Do you think we can turn off these lights? Or
I'll try another
Yeah.
configuration.
I'll-
Totally dark here, that's not.
Okay, that might be.
I think that's what we had initially, isn't it?
I think that's.
Hello everyone.
Okay, that looks great.
Hello everyone,
Okay. Just gonna let it go.
and welcome. My name is Tadhg. I am a postdoc in the Kerry lab and the Thomas lab, and today I'm going to talk to you today about my research, which is regards to setting a roadmap for integrating ecological forecasting into undergraduate ecology curriculum. There we go. An outline of what I'm going to talk about today is I'm going to give a bit of background to the Macrosystems EDI project. I'm going to go into why ecological forecasting, give you a bit of information in regards to how we collect our data, talk through some of the results and discussion that I have so far, and then a bit of a quick overview of some of our future plans. And here's a photo of all the people involved in the Macrosystems EDI team. So, Macrosystems EDI is a project where we are building modules that challenge students to confront ecological complexity by incorporating drivers and responses across multiple scales, while building quantitative skills through the R programming language and also through Shiny apps. Each Macrosystems EDI module is designed around specific student learning objectives, and these objectives cover aspects of all 4 dimensions of ecology education, and use the 4-dimensional ecology education, the 4DEE framework. Students learn core ecology concepts, address systems thinking, they're allowed to develop and test their own hypotheses about different things like ecological forecasting or land use or climate change, effects of climate change. And then they also gain computational skills through hands-on engagement of, with module materials. Here's a list of the modules previously developed within Macrosystems EDI by postdocs former postdocs in the Kerry Lab, Alex Hounshell and Kate Farrell. And each, what an EDI module is, it includes the instructor lesson plans, pre-class readings, in-class activities, with homework questions, with answer keys for the instructors. And all of this can be downloaded as is from our website, and it can be used as is in any kind of class, or it can be, or the materials can be modified to fit into a particular class needs. So here's some of the previous work that have been done, as I mentioned, from these modules. So a couple of publications from, this one's from Alex Hounshell et al. And they found that students were more likely to be able to correctly define macrosystems concepts, apply macrosystems approaches, and to be able to interpret complex nonlinear ecological processes. And then also there's a publication by Kerry et al., where from those 4 modules they found that yes, students were able to gain confidence in their ability to use ecosystem modules, and they found it was a great way to introduce undergraduate and graduate students to ecosystem simulation modeling. So the question then is, why ecological forecasting? Well, ecological forecasting itself is an emerging field, and it can be used to improve understanding and allow us to predict changes in ecosystems. These forecasts themselves allow people to prepare for events, and then it also allows for actions to be taken to prevent undesirable outcomes. And there are many aspects, dimensions within ecological forecasting, from creating a hypothesis through collecting data to building a model and generating your own forecast. And ecological forecasting cuts across all these different fields. Some of the gaps that we have identified in ecological forecasting education is that most of these materials have been developed for masters and PhD students, and our main motivation for this project is to introduce ecological forecasting to undergraduate students by developing hands-on and interactive materials that are accessible for both the students and the instructors, because we recognize that there can be barriers there for instructors as well. So here's a list of the 4 modules that we're developing in this, as part of this grant. I will be talking about Module 5 particularly today, which we've developed and tested last spring, and Module 8 has been developed and tested by a PhD student, Whitney Wimmer in the Kerry Lab. So our kind of central hypothesis, or here today is kind of, can we use short hands-on modules with or Shiny apps to introduce and engage students into ecological forecasting? And just give you a quick overview about how we collect their data. Students are given a pre-module questionnaire. Then the module is taught. The instructor gives students some pre-class activities. Then there's a 20-minute introductory lecture. And then it's a 1.5 to 2 hour activity with facilitated discussions. And then following completion of the module, students then complete a post-questionnaire, and then we compare the student responses in the post-questionnaire with the pre-questionnaire to assess students' knowledge and growth from completion of the module. So some of the things we collect are students do some self-assessment of their proficiency and confidence with regards to certain aspects of quantitative ecology. We question their understanding of some of the core ecological forecasting concepts, which are within the materials, and then also the knowledge that they have gained in kind of questions of with regards to advantages and disadvantages, or advantages and challenges within ecological forecasting. And some of the data I'm presenting is from 6 classes which we had in the spring. We taught this module, and we have 3 graduate and 3 undergraduate classes. So just to give a quick overview of what a Shiny app is it's an interactive web page built using R, and this is kind of to get over that intimidation barrier for students and instructors when kind of working with ecosystem modeling. And this allows students to kind of engage in the task, complete tasks, answer questions, and run different models or different forecasts within one interface. Some of the results I'm going to show is from these questions that were in the questionnaires, where we asked students to rank their proficiency and also their confidence with regards to generating ecological forecasts. We used the 5-point likelihood scale questions, where students where they go from no proficiency or not at all confident to expert proficiency and very confident. And here are the results. We've faceted them on by proficiency and confidence, proficiency and top confidence at the bottom. On the left is graduate students, on the right is undergraduate students. On the x-axis is the students' response on the pre-module, and then on the y-axis is the change after the module. So what was the difference? So for example, with the 1 here, you'll see that students who went from, had an increase of 1, they went from 1 to 2 in terms of their proficiency. And overall, we saw students who ranked themselves quite low in terms of confidence and proficiency across both undergrad and graduate students. They saw the largest gains in terms of after completion of the module. And this is with regards to generating an ecological forecast. We also asked them with regards to communicating the output from an ecological forecast. So similar 5-point likelihood scale, and we saw very similar responses where students who had low proficiency, low confidence in the 1s and 2s on the pre-module, they saw the largest growth. And it's quite interesting as well how undergraduates tend to have a larger growth there. I guess kind of grad students, you could talk about imposter syndrome or things that maybe understanding more nuance, but this is just from our 3 classes, 3 undergraduate and 3 graduate student classes that we have so far. One of the questions, we asked students to pretend that they're a forecaster, and they have to, what steps would they propose to accomplish the goal to set up a forecasting system. So like I said, we introduced this forecast cycle that students step through in the lecture, so we were looking for students to kind of use these, identify these and include these in their answers. And here are the results, where on the y-axis we have percentage of students who included that answer, and the answers are on the x-axis. And what we can kind of clearly see is that we did see like increases from pre to post for both graduate and undergraduate students across these ones here, in terms of kind of some of the more central ideas with regards to ecological forecasting. And then some of the more advanced topics, such as uncertainty and updating forecasts or data assimilation, we saw greater increases for graduate students, but we see that these are, there's increases still nonetheless. And then one of the other questions we asked was, we asked students for listing some of the challenges of ecological forecasting. And what was really interesting here was, while again we saw increases generally in the post, but one of the ones that students outlined was modeling, or anything to do with ecological models, and then also uncertainty, which is given us kind of a great impetus, because the next module that we're developing is focused in on ecological forecast uncertainty. So kind of some of the main points that we got from this is that kind of students with most to gain the most. Like I said, students who rank their confidence or proficiency quite low in the pre-module, they are the ones who see the largest increase. That this module so far has proven to be valuable in increasing both undergrad and graduate student understanding. These orange shiny apps help to reduce that intimidation barrier when teaching some of these complex concepts, and introducing students to these on a higher level. And that's stepping through the entire forecast cycle itself. So students do that in the materials, and this kind of improves the students and gives them a greater understanding of the steps involved in. There we go, forgot the animations. And yeah, so some of the future work is like I highlighted there on the last slide, where we're gonna, well, first of all, I'm gonna be writing up this data kind of into a publication with the idea of using this as a setting out that roadmap for why ecological forecasting should be included. We're also coordinating and collecting more data this fall semester, and then also developing this next module about ecological forecast uncertainty. And the results from the first module is really promising, and showing that this is an area that we can tap into and provide as a follow-up education material to the first module. I'd just like to say thank you to funders, NSF, Microsystems Biology Grant, which funds the work in this project, my Project Eddie colleagues who are listed here, Keelan Carey, Quinn Thomas, and Whitney Ulmer, and then all the personnel involved and also the data providers. Anybody have any questions?
If we ask a question, will he answer it?
It could happen, sure. I could type it, I think he can hear us. So yes, you can ask a question and I will tell you his answer or I'll make something up. Pretend it's working.
I could potentially also assist one of the also creators of some of the EE modules, if you can't answer.
But does anyone have a question?
Just Joel's hypothetical one.
Just meta questions today. Okay.
You ready, Whitney?
So it looks like that the undergraduates, even though they have more confidence, they're not using what you gave them in their forecast. Because it looked like they're still lower where the graduate students were using more of like the data and uncertainty and everything in the forecast challenge. Is there a way, why do you think that is, and is there a way to kind of get the undergraduates to use the tools that they've learned about?
Are you talking about the figure where he showed some of the
Pre
layers?
the
that
pre
they
and the
were-
post, yeah.
It could be something where they were just less familiar with that idea before. That one's a harder question for me to answer because it's more specific for the modules.
Okay.
He's typing.
Oh. Should I just ask my question?
No, go ahead. We'll
Okay.
just overlap them.
So my question was about essentially the slope of that line, such that you had people who felt very confident and then took the module and then felt less confident. And I was wondering what you thought the reason for that was. Yeah, I think that's been common throughout the modules, and not just this one, but the four that he mentioned at the beginning that have been around for more years and been tested more. And I think, like Tadhg mentioned, imposter syndrome is something, I think once you get more familiarity with something, you also realize that there's more things you don't understand about it. And that's kind of an interesting result that seems counterintuitive and doesn't exactly support the fact that these modules are, it's not the result we would look for, but I think it actually highlights the ability of the module to expand the knowledge that students have in that topic.
Okay, answer to the first question from Tadhg. Good question. We find that undergraduate students find this material a bit more complex, so hopefully with more data we'll see more benefits. Should we go ahead and change it over, and if an answer to your question comes in, Kate, I'll relay it.
I find it amazing that anyone would give this sort of 5. At the beginning, like, I'm an expert at this.
Yeah.
Oh, never mind.
So, maybe they just were confused
Oh, yeah.
about which way to scale with.
I mean, it seems like progress if they go from a 5 down, right? So.
What did you notes?
No.
You good? Okay. I'm gonna close the door.
Okay, thanks for waiting a second. So we'll have Loren up next. Please go ahead.
Okay. Hello. So good to see people, like in an auditorium. I mean, unfortunately, we're still wearing masks, but it's better than not seeing people in person. So I'm Loren Vega. I'm a new-ish member of the Whitehead Lab. I joined February of this year. And so today I'm going to be talking to you a little bit about what I've been doing this summer, basically. So the project that I was hired for and that I've started to dig into and mostly see if you guys have suggestions for where to go from here. So when you think of an apple, can you still hear me there? when you think of an apple, or at least what I used to think of when I would think of an apple, was this really red, delicious, usually spotless fruit that we always find at the stores, right? I grew up in Latin America. Apples are not very common, so when I used to get them, my grandpa used to sell them, apples and grapes, which are not tropical fruit. So it reminds me a lot of him, and I would always picture this awesomeness. But something we do not think about is that they're covered in microbes, right? Not only outside, but also inside. And usually we think about them as all the pathogens that might be there. So if it's nice and red and waxy, because it's being waxed, it means it doesn't have any of that. But the truth is, as everything in our environment and ourselves, all these microorganisms are present there. And so I found it very interesting when Suzanne talked about trying to look more into the microbiome of the fruit. We also, I'm an entomologist by training, so so I usually think about microbes in the leaves, in the roots, in the soil, but not so much in the fruit, or even inside of the fruit. So this was very interesting. Actually, in 2019, I was like, oh, last year. No, never mind, that was like 2 years ago, but last year, I think we're not counting it. Wasserman published this paper where they actually grouped apples in a conventional, certified conventional according to European laws versus certified organic. And what they saw was that there's big differences between, he focused on bacteria in this case, and he noticed, they noticed differences in the diversity of the apple based on organic and conventional. But most interesting was that they noticed differences around the apple, like within the apple. So the stem is different than the stem end of the apple, which is different than the peel, the calyx, the fruit, the pulp, which is very interesting to us. Sometimes we don't think that such a small thing can have such a diversity, especially when it's being grown the same way. So we thought about looking a little bit more into this, or Susan thought about looking a little more into this. And some of the reasons why it is very important for us to look into microbiome in fruit is that all these microbes or microorganisms could be changing the nutrient availability in the apple. It can also affect the secondary chemistry within the apple. So the phenolics in this case is usually where we're very interested with apples. Most importantly, and why this organic versus conventional idea is interesting is because it can be competing with pathogens. So when we're applying all these different management methods, we could be affecting actually the apple's own way of defending itself. It can also protect the apple against herbivores. And in the bigger picture, if we try to understand all these mechanisms and factors and everything that's going on in here, we could say our broad impact, or what we're looking for, is to be able to maybe develop some more sustainable methods of pest control, and even human health. Apples, and 1 apple a day keeps the doctor away, and so on. Now let me tell you, I told you about how excited I was to work with apples, how many good memories I have of apples. I don't eat apples since I started working with apples, and that is because they get sprayed so much. It is, I have no idea. And I've always, you look into the dirty dozen and you're like, oh, apples are there. And you're like, whatever, spray some water and eat them. And so I've stopped because it gets sprayed so much. And so if we're able to specifically find ways to lower those sprays, it would be great for everybody, not just the farmers, economically, the environment, and definitely ourselves. And then maybe I can get to enjoy an apple pie more now than what I do right now. So some of the things we were trying to tackle this summer were what factors influence the microbiome and how, and there are many, okay? A lot of them. So for this summer, we focused, number 1, in management practices. So we saw the paper where they did conventional versus organic They focus on the entire management of it, and also they had a pretty low sample size. So in our case, we're only focusing on fungicides, and that is partly because it's one of the most sprayed pesticides for apples. They're attacked by a lot of different fungal pathogens, and so they're sprayed quite a bit against that. And so first we thought about doing it throughout the season. So because apples, especially here in the East, are under a lot of pest pressure, there was regular management throughout the entire orchard. So there's a new orchard, for those of you who don't know, newish. This is the first season that it's producing fruit in Kentland, and so for the first few years and all throughout the flowering, the management of the entire orchard was the same. So very conventional, what you would expect in a commercial orchard kind of thing, because it is under such high pressure of pests. If we were not to do it the same, we probably wouldn't have apple in our controls. So once flowering happened, we split our orchard. In our case, we focus on the gold rush. There's a lot of genotypes, but we focus only on gold rush. We split into 6 treatments. That was a control, which would only spray water, 3 conventional pesticides, Captain, Inspired, and Flint, and then 2 organic pesticides or fungicides, copper and sulfur. And then we would do sprays every 2 weeks, which is more or less what is done in a commercial orchard. And then we would collect samples. I collected samples before the first spray, so kind of have an idea what all the apples had before we started spraying. And then we're gonna have a final collection during harvest, which is happening hopefully sometime in September. And the samples that I've been collecting are of the surface of the apple, the peel or the skin of the apple, and the pulp. And we're taking samples for metabolomics analyses and phenolics, as well as some DNA sequencing to actually see what microbes are in there. And we added a third thing about trying to start establishing a yeast library. So we decided to focus, instead of bacteria, focus on yeast and start developing and looking into like what culturable yeasts are present in these apples. So because like what we saw previously, the microbiome changes a lot throughout the apple. We focus only on basically the equator of the apple. So that's where we collect a lot of samples from to kind of reduce a little bit of all that variability that we're going to have if we swap everything. And like I said, we did surface, peel, and pulp. The other thing we also wanted to look at is shorter term turnover of the microbiome and the plant chemistry in the apple. So for this, it's just kind of looking between the last spray and 2 weeks after that. So took some samples 2 days before the last spray. That was last week. You'll see soon why I'm so excited about this. Then we took sample 1 day after the last spray, 7 days, that's tomorrow, and then 14 days, that'll be next week. And it's the same processing that we did for the other ones. Phenolics, metabolomics, DNA sequencing, and the yeast library. And so right now we're still, like I said, I'm still collecting samples, so I don't have lots of data or anything like that. However, we did start, never mind. And finally, it sounds like a lot of stuff for one summer, but finally, the other thing we're gonna be looking at, the other factor other than management, is just the apple genotype. What are the differences in the microbiome and the plant chemistry just due to genotype. Now, there are about 18 different genotypes in the orchard right now. However, not all of them are fruiting. So next week, I'm going to be going in there and just basically collecting apples from every tree that's fruiting that's not in my other experiment. And so then we'll see actually how many different genotypes we're going to be able to test for, and again, it's very similar processing of it. So I'm very excited about this, and mostly I'm very excited to figure out which ones we're even going to be able to sample from. So anyway, like I was saying, I still do not have results of all the massive amount of sampling that I've been doing, but we do have some initial data of yeast that are present in the apple, and these are from the samples that I collected before any spray occurred. And then I have some more yeast growing right now from all the other ones. So we're very excited to see what had happened as the season went through, what other geese might have shown up. So we have some Metskinikovia. I'm practicing so hard. And Chrysoperle and Pulcherima. These are known to be involved in winemaking. So Susan and I are very excited because we also love cider. And so we're looking forward to see how important this might be to identifying some cool cider, new cider yeast. But also those three and some others are involved as biocontrols. And like I mentioned previously, that is like one of our ultimate goals is identifying other things. So this is very exciting about going forward and maybe doing some testing and see if there actually has some biocontrol potential against the bud rots and all the other fungal pathogens that apples are exposed to. And like I said, these are from the early season collection. And so this is, in case you cannot tell, I'm the short one here. And Susan, this was us ready before our very first spray. And so every spray in this awesome humidity of Virginia and heat required this full Tadhg suit, which is why I'm so excited that I'm done spraying. I picked the project, so I cannot complain. But if you have questions or any kind of suggestions, especially for analyzing the massive amount of metabolomic data that's coming my way, I would highly appreciate it. So.
Okay, I think we have time for one question.
Yes, Emily.
Yeah, so I guess with the like different, like are you looking at diversity again, maybe as like one of the predictors?
That will hopefully be one of the things we see.
I guess if you were like, what would your prediction be between sprays? Like what do you expect to see?
Oh, that's a very good question. So I would probably expect to see, based on just how the apples look like right now in the orchard, probably a little more diversity of microbes in the organic ones. They're just not looking as clean and nice as the conventionally sprayed ones. So, but that's just like external, right? Surface stuff. One of the other things I noticed from like the yeast thing, the yeast that we have right now is that the yeast were isolated from surface, skin, and pulp were not the same. So all those different ones came from like different parts of the apple. So hopefully we'll also see that again throughout the treatments as the season went through. Thanks.
Okay, thank you. Okay, so we'll just have one more, which is, I think, pretty short and is also pre-recorded.
While we're getting that loaded, we'll have 4 more next week. 3 more? 4 more? Can't remember.
4 more, yes.
4 more next week here, Postdoc Flash Talks. And I think most of those people will be here.
We'll see.
We'll see.
We thought everyone would be here today, but you know, things are coming up, so. Okay. Okay.
Hi, I'm so sorry I couldn't be with y'all today, but I'm very excited to share the research I've been doing at Virginia Tech with the Walters Met Lab and the MIMS Lab, covering how we use niches to inform conservation. So a niche describes how animals vary in their use of resources. In my past research, I've used freshwater mussels along a drought tolerance gradient. But you can also consider how different animals use different food resources or maybe temporally separate. Climate change and land use are drastically altering the niches available in a landscape, which spells bad news because the loss of a niche often leads to a loss of that animal, because without those resources, the animal ceases to exist. In my flash talk today, I'm talking about two different studies. One is investigating how frogs and toads exist along a climate gradient and how they vary in their sensitivity to climate change based on the breadth of climates they exist in. And then the second part is looking at how red-cockaded woodpecker management affects the availability of different niches in a marine camp base, and how bird assemblages react to that different availability of niches. But overall, I just wanted to give you a sense of who I am as an ecologist. I study animal assemblages and the variation in the niches those animals occupy to better understand how animals exist and hopefully continue to exist in the future. So with the MIMS lab, I am ranking climate change sensitivity for frogs and toads based on the climate niche breadth, or the variation in climates those frogs exist in. And so to get that variation and measure sensitivity, we use publicly available point occurrence data, like you can find in GBIF or HertMapper if you write a good proposal. We then pair these point occurrences with open access climate rasters to ultimately build an idea of the climate niche that this frog exists in, such as in Axis stabilis. So here we can see the different variation in climates, precipitation this frog exists in. We can do this for a bunch of different species. And essentially, this puts a lot of species on the same scale. For example, on this gradient to the right, we have precipitation on the x-axis and all the species we analyzed on the y-axis, with precipitation mean values being the dots, and then the line representing the standard deviation of the climates those point occurrences were paired with. And so we can compare the western toad with its very large climate variance with the Pacific tree frog with its moderate variance, and then the southern leopard frog, which has very small climate niche breadth for precipitation. We do this you can do it for any number of climate variables, but we did it for 3 variables, maximum temperature and minimum temperature it exists in. And you can build this ranking of climate sensitivity, because you can imagine that smaller niche breadth might be more sensitive to climate change than the one that has a wide variation in climates it exists in. And so what we find when we combine all 3 of these climate niche breadths measurements is we can then rank species based on that sensitivity, where the frog, the southern leopard frog that had that very small climate niche breadth for precipitation, is more sensitive to climate change than the Pacific tree frog, which is less sensitive than the western toad. And ultimately, this allows us to identify frogs that might be flying under the radar or might be missing from the conservation picture and might warrant more attention.
We had slide 7.
Yeah, it's pretty frozen.
Try this. Yeah, it looks like it. Yeah, it's just like not responding at all.
Yeah. Sorry, I don't know how to use PC.
Me neither.
Okay.
No, that's not doing anything.
Yeah, I think it was in the 90s. I'm not sure if it still is. Oh, okay. Thank you. Okay, thanks for coming, everybody. I guess that's it for today. We're gonna do better next week.
And if any of the grad students and postdocs wanna introduce me, those people who are new or anything could gather just outside the front, not in the atrium, but outside maybe. I know there's some new students in the audience and new postdocs. Maybe we could just do some introductions of those once we figure out how to turn off the computer. But otherwise, thanks for a difficult first day. We're happy to see all of you.
Just what I told my class. I'll just turn the screen off so at least people can't see what we're
Crashed it.
that doesn't sound like the most reliable thing.
No.
Are we at least not streaming anymore?
Oh, that's a good. Doesn't look like. Oh wait, yeah.
Show streaming